Clear? At the time of gamete formation, crossing over takes place between these two genes. Gene 1 reaches to one gamete, gene 2 reaches is one gamete. Here also crossing over takes place, gene 1 reaches one gamete, gene 2 reaches one gamete. Here crossing over chances there are very less, because genes are, the linkage is very strong. If bond is very strong, you can able to break? No. If bond is very weak, if you able to break? Yes. Suppose for example, two friends are there, two close friends. If bond is very strong between those two people, anybody can break that bondage. Anybody can break the bond between them? No. But bond is weak, obviously anybody can break, and third person will interfere. You understood or not? Here also same concept you should remember. And we are saying some of the friends, thickest friends also there, right? Okay. See here, if crossing over takes place between two linked genes, then the genes get segregated and they will assort independent to each other. When crossing over takes place between two linked genes, then the genes get segregated and they will assort to the independent to each other and reaches to the gametes. But there is no crossing over between these two genes. There is no segregation of genes. Whenever crossing over occurs, then only segregation, separation of genes takes place. But there is no crossing over between these two genes. There is no segregation of the genes. Gene segregation is not takes place. Hence only parental combination will be found in gametes. The same genes or reaches, the genes are not separated, the linked genes are not separated, and same genes are reaches to the gametes. Then what happens? Whenever genes are not going to be separate, obviously parental combination will be found in gametes. You got it or not? When both genes are separated, and whenever the both genes are separated, then only there is a chance for new variety. But linked genes are presented, but there is no crossing over between these two genes. There is no segregation of genes. Whenever there is segregation of genes is not takes place, then what happens? Hence only parental combination will be found in gametes. More parental combination will be found, less recombinant varieties are formed. You got it or not? Right. See, this 2 points you should remember. This was first discovered, linkage first discovered by Batson and Punnett, 1906. Lathyrus odoratus, sweet pea plants. T.H. Morgan was the first to relate linkage to the segregation of homologous chromosomes. If crossing over takes place between two linked genes, then the genes get segregated. They will assort independent to each other when crossing over takes place. But there is no crossing over between these two genes. There is no segregation of the genes. Hence only parent in combination will be found in gametes. If crossing over is there, segregation of genes. If crossing over is not there, there is no segregation of genes. And both the genes are reaches to the same gamete, and parental combination will be found in gametes. Clearer? Right. Linkage group. Next concept, linkage group. All the genes on one chromosome which remain together during heredity transmission are said to form a linkage group. All the genes on the one chromosome which remain together during heredity transmission are said to form a linkage group. The genes are presented, all the genes are presented in one chromosome which remain together during heredity, means transmission from one generation to another generation, are said to be a linkage group. Right? Linkage group, same group. The number of linkage group in an organism is equal to haploid number of chromosome plus the number of kinds of allosomes of the organisms. The number of linkage groups in an organism is equal to haploid number of chromosome plus number of kinds of allosomes of the organism. For example, in humans, how many chromosomes are presented? 23 pairs or 46, right? So how many linkage groups are presented in humans means the linkage groups in an organism equal to haploid number of chromosome. In human, diploid number of chromosome means 46, haploid number of chromosome means 23, plus the number of kinds of allosomes of the organisms. Allosomes means sex chromosomes. In human, there is two types of sex chromosomes are presented, X type and Y type. So X presented in female, Y presented in male, means XX presented in female, XY presented in males. In humans, in human males, two types of allosomes, X type allosome, Y type allosome. In human females, only one type of allosome, only X type of allosome. You got it or not? Now, the linkage groups in human females, how many? 23 linkage groups only. Why? Because the number of linkage groups in an organism equal to the haploid number of chromosomes, 23 chromosomes, haploid number of chromosomes, plus, according to this definition only we will go, plus number of kinds of allosomes of the organisms. Number of kinds of allosomes, how many? Only one type. So 23 linkage groups only. But human males, number of haploid number of chromosomes, haploid number of chromosomes, how many? 23, plus, and the number of kinds of allosomes, how many? Two types, X and Y type. Y type is extra, 23 plus 1. Y chromosome has different set of genes as compared to X. So 23 plus 1, 24. You got it or not? Why here not taken 1? Why here taken 1? Because human females linkage groups, 23 linkage groups only. Because all are same type of chromosome. We should not take 23 plus 0. Why? Because all are same type of allosomes. Here what given? The number of kinds of allosomes. Suppose 3 are presented. 23 plus 3. 3 not presented in humans. No need. So human males linkage groups, how many? 23 plus the kinds of allosomes, how many? Y chromosome extra is there. So 23 plus 1, 24 linkage groups. Understood or not, right? So linkage groups, all the genes on the chromosome, on the one chromosome which remain together during hereditary transmission are said to form a linkage group. The number of linkage group in an organism is equal to the haploid number of chromosome plus the number of kinds of allosomes of the organism. The haploid number of chromosomes plus the number of kinds of allosomes of the organism. Human females, 23 linkage groups. Human males, 24 linkage group. 23 plus 1. Because females has only one type. Means all are X type only. But males, X and Y. Okay? Right. This is regarding about linkage and linkage groups. So next one is recombination. It is the reshuffling of parental genes to produce new genotypes or new combination of genes. It is the reshuffling of parental genes to produce new genotypes or new combination of genes. Recombination means what? Recombination. Whenever at the time of gamete formation from the parents, what happens here? At the time